India, China, and Indian subcontinent in particular, and some African countries. But recent years, emergence of multi-drug resistant tuberculosis, and particularly tuberculosis with HIV co-infections, caused a major concern in the developed countries as well. So there is an urgent need to develop some new antibiotics to circumvent the problem of multi-drug resistant tuberculosis or HIV co-infection with tuberculosis. Among the treatment available for tuberculosis, there is a vaccination available, BCG vaccination, discovered in Institut Pasteur Paris during the time of Second World War, and it was given for it is given for infants, but it is not reliable for adult pulmonary TB. Here comes the role of antibiotics as a part of multi-drug therapy. Normally, isoniazid, pyrazinamide, fatty acid biosynthesis inhibitors, rifampin, RNA synthesis inhibitors, either ethambutol, which is arabinogalactan and peptidoglycan synthesis inhibitor, or streptomycin, a set of 4 drugs normally used for a period of 4 months, and then 2 months treatment with isoniazid and rifampicin. But this prolonged treatment causes severe side effect of hepatotoxicity, which forces the premature treatment termination. And in association with this premature treatment termination, because this is a long treatment time, there is quite a lack of treatment observance as well. So, which allows the bacteria to mutate and develop resistance to these drugs, causing multi-drug resistant or extensive drug resistant tuberculosis. Now, if we see among the drugs employed normally for treatment of tuberculosis, we find that majority of the drugs like isoniazid, pyrazinamide, or ethambutol, these are all targeting cell wall biosynthesis of bacteria. If we take a close look at the cell wall of constituents and the cell wall of bacteria, Mycobacterium tuberculosis, we find that these are mucolic acids, which are major component of bacterial cell wall. And fatty acid synthesis 2 cycle, this is an integral part of mycolic acid biosynthesis. Importantly, fatty acid synthesis 2 cycle is an exclusive feature of prokaryotes. So targeting this fatty acid synthesis cycle is quite advantageous in that respect. And if we take a close look into the different intermediates in the FAST2 cycle, we find that there is an alpha-beta enoyl acid, alpha-beta unsaturated carbonyl thioester, or enoyl acid thioester moiety is there. We can naturally find that in cinnamic acid. Cinnamic acid is an intermediate in the formation of lignin from phenylalanine in plants, and it has low toxicity in human exposure as well. But question may be asked, why particularly cinnamic acid? There are other alpha-beta unsaturated carboxylic acids in the nature as well. Cinnamic acid has a long history of treatment in tuberculosis treatment. In 1894, Warbus reported the successful use of cinnamic acid to the tuberculosis patient. In 1920s, ethyl cinnamate, sodium cinnamate, and benzyl cinnamates were also used to treat tuberculosis. In recent years, it has been shown that cinnamic acid is actually not bactericidal. It was shown by Stokke et al. And in their experiment, they also shown that ofloxacin or amikacin or glufazimine, all these are established antibiotics. When treated in combination with cinnamic acid, this combination eventually showed sub-MIC inhibition. MIC is the minimum inhibitory concentration of a compound on bacterial cell growth, which is visible growth actually. And on this strain particularly, this is a drug-resistant strain. So there is an we plan to modify and particularly of this compound, where the introduction of the cinnamyl moiety to the rifampicin molecule actually enhances biological or anti-tubercular activity. So these two particular reports were very much gave us the boost to design something on cinnamic acid. So we wanted to functionalize cinnamic acid in this way. We wanted to control the lipophilicity by production of good alkoxy substitution at para position and attach some drug-like molecule in the carboxylic functionality. Different alkyl groups have been chosen to control the lipophilicity, but why lipophilicity is important? Because bacterial cell wall is greasy, and according to Lipinski, lipophilicity, which is expressed in C log P, should be between 2 to 5 in order to have a good penetration for a druggable candidate. And we chose to attach a different substituent to the acid functionality. Thioester we targeted as thioester in order to have a resemblance with the enoyl acyl thioacide carrier protein. But we also realized that thioester could be, because of carbon sulfur weak bond energy, thioester could be fragile under physiological condition as well. Therefore, we also plan to introduce its nitrogen variety with ethylene diamine moiety. We chose to have an aromatic amide as well. Isoniazid is an unknown antitubercular drug. Hydrolazine is antihypertensive drug. And this triazolothalazine particularly obtained serendipitously just by hitting the hydroxide derivatives. This is how we made different targets. Just very simple, by taking alkoxy cinnamic acid and attaching different heteroatomic as well as different hydroxides and amides using peptide, normal peptide coupling. This triazolothalazine was initially obtained by heating its hydrolazine derivative, but later on we made it work in one pore sequence. So, among the synthesized compounds, first I must tell that we assess our biological activities with 4 parameters. MIC, which is a minimum inhibitory concentration for visible growth of MTB. Cytotoxicity, which is a measurement of lethality on healthy human cell. And these two are experimental parameters. And C log P and PSA polar surface area, which is a measure of possible binding area for a protein to the molecule. And these two are theoretical values. So for this thioester series, this geranyl derivative was the most active with MIC value 1.5, but its cytotoxicity profile is not so good. Selectivity index SI, which is a ratio between IC50 and MIC, which should be more than 100 to be a good druggable candidate, is only 23. So its cytotoxicity profile was very poor. We had to abandon this series of compounds for further medicinal or biological studies. This ethylene diamine compound derivative was the most active in its own series, and it has got a better cytotoxicity profile with SI more than 100, but it's not better than isoniazid, which is a reference compound. However, this 2-amino